Hello everyone, welcome to AstroStream. We have Reut with us talking about astrobiology, which is really interesting topic, and Reut is an expert in it. She also has PhD, and today is our third episode out of total of 6 episodes of this series. So today we are going to talk about the life and evolution in the universe. I hope you guys are enjoying the series so far. And also, if you want to have a certificate for this course, we are gonna give you the certificates after the whole series is done. So there is a form link in the description of the video. You can fill that up so that we know that you're watching the videos, and we can send you the certificate. If you have any questions, do post them in the live chat. Reut is also watching them. She says hello and good evening in the chat if you can see her. So post your questions, and definitely she'll answer them at the end of the stream. Again, like last time, it might happen that the connection or some technical problems would occur, but I hope they don't. So just bear with us, stay tuned, and we are going to try our best to make this experience great for you. So now off to Reut.
Hello. Thank you very much. Hello, everybody. Good evening. Good afternoon to my friends from Israel, and good evening to my friends from India and from wherever you are. Welcome to my astrobiology series together with AstronEra. This is exciting times. I was very happy with your question and enthusiasm last time, and I hope you will enjoy this one as well. Let's start it with my presentation. And if you have any question, Shveta is watching the YouTube as well. She's looking at your comments. At the end, we're going to find and we're going to answer a few of your comments. And also, I'm very happy to tell you that what's going on? Yeah, is it happening? I don't know. Let's see.
Yes, we can see your screen now.
Can you see the presentation now?
Yeah.
Thank you. Okay. So what I want to say is that at the end of this presentation, there's a very cute riddle. Okay. So it's well worth for you to stay till the end, and Shveta will decide whoever, solves this riddle first, whatever prize they get. I don't know. That's not my part, but I'm just saying there's a very cute riddle at the end of this presentation. It's well worth your time to stay. So during this course, we have 5 meetings. This is the 3rd meeting. At the first meeting, which you can see online at YouTube and in watching your own time, we talked a little bit about introductory, what is astrobiology, who am I, who are you, what are we doing here in the universe? And last week, we talked about the beginning of life. So when I talk about astrobiology, I usually cover all these different topics, starting from the origin of life, how life started on planet Earth, what type of evolution did we go through, what are the atomic and organic elements that we see in the universe, in the galaxy, in the solar system, how does that affect life here on Earth and perhaps on other worlds, how did life begin? There are different theories, and we spoke about it in the former lesson that we just had. We talked about autocatalytic sets, we talked about the lipids world, we talked about RNA world, we talked about primordial soup, how did we prove, how scientists have proven that amino acids can be created in an abiotic fashion without any life. And we talked about whether life have originated perhaps in hydrothermal vents, perhaps underneath the oceans, perhaps as, stromatolites in very shallow bays. We talked many things in our last session, and then if you want to go and have a look there, you're more than welcome. Today, my talk is going to be more about the evolution of life and some very, very basic concepts about the evolution on planet Earth. We actually know quite a bit now. And I actually found out that Wikipedia has an amazing series about about life evolution and biodiversity. And it's covered very, very thoroughly in Wikipedia. And if you're into that, you're more than welcome to go and check it afterwards, after my presentation. And I'm going to talk a little bit today about the timescales that are involved in all this big topic on how life has emerged. How long does it take to do an evolution? What is evolution? So today, like I said, we're talking about life and evolution in our astrobiology, which is life center, multidisciplinary science. So what we're going to talk about today is what is life, and we have to go back on this. I know we covered some part of this topic in our recent encounter, but this is very much relevant for this presentation as well. And why does life need evolution anyhow? We need to answer this very basic question. Does life need evolution? Why does it need it? Let's think about it a little bit more after I give you a bit more background. What is evolution and how we understand it today? How long does it take to evolve or to devolve? Sometimes some things, they go through a process in life and they go in one direction, but then they can very well go into a completely different direction or go backwards and try a different type of an ecological niche. So how long might that take? And we know so many life forms here on planet Earth. And last but not least, our favorite topic is, what about other worlds? Can life and evolution, as we know from planet Earth, did it happen someplace else? Do we know? What do we think? What do we think about it? So this is our topics for today. I'll try to talk slowly. I'll try to talk clearly. I hope everything will be clear, and feel free to that as we go through the presentation. And if you have any kind of questions, write it down, and then at the end of the presentation, me and Shweta will start answering your question in an orderly fashion. So some of you might know, but I don't know, some of you might not. Can you guess for 5 seconds, what am I holding in my hand? It's a very interesting thing. I did it in my PhD. It's from Shark Bay in Western Australia. So these wonderful things are actually biological in origin. It looks like rocks, and it has carbon and carbonates inside it. But the whole thing was structured by a lot of microbial activity. And this is one of the well-known formation of stromatolites that live today in Shark Bay in Western Australia. Now, as you can see, there are different layers. So if I'm talking, for instance, on the upper left picture, where you have all these gray rocks, and above it you have a green, yellow, beige. Basically what you have here is that on top you have photosynthetic microorganism who can do photosynthesis from the sun. And underneath it, you basically have sulfate reducers, microbes. It does look like a rock, but it smells awful, like stinking eggs. And when you look down on the left, my left lower, and I'm holding in my hand something which is very gooey, that's like a biopolysaccharide. They issue these things and they form like a biofilm within these stromatolites. And what it helps them to be coherent and to stay in the same form that they want when they're surrounded in basically in liquid water, because the waves come and go, they can wash them. So this polysaccharide, this gooey stuff, and we find it in other formation on planet Earth, it also protects them from salts and from too much, water washing them away. So what we see here is the intermix between microbiology and life forms and rocks. And what's interesting about this is that we now think that this was the earliest life form, this type of microorganism lying upon one another, doing this exopolysaccharide gooey things and forming rocks. This was basically perhaps how life has begun on planet Earth about 3.5 to 3.7 billion years ago. We find these squiggly lines in really, really ancient rocks. This is an example from Western Australia, from the Pilbara region up the north. And we have geologists confirming that these shapes, they are called stromatolites. Stroma means spreading, and from Latin litos, which is stone. And we see these stromatolites, and we see these type of microbial, it's not microbial shapes, but this shapes inside the rocks. They come from places that had water before, perhaps billions of years ago. And like I've shown you before, we actually find this type of living, this geobiological living, we find it even today in Shark Bay in Western Australia, and we find it in really, really ancient rocks. So this is how we think life perhaps have begun, and this is the best evidence we have because it comes from really ancient rocks. And Earth is about 4.5 billion years old. So if we go by this dating, then life has begun on planet Earth almost 1 billion years after it was formed. That's rather quick. It could be even just 500 million, 500 million years. So this is an interesting point to start our discussion about life before we go into evolution. When we look at evolutionary timescale on planet Earth, what we see here is an interesting graph. What I want to show you, let's focus first on understanding this graph. So what we have here is we have a horizontal line, which is the x-axis, and we have a vertical line, which is the y-axis. The vertical line goes from 0 up to 900 and states the number of families. And it means the number of families of species, the family of life forms. How many families? The number of families in millions, basically, or thousands, 900,000, 600,000, and so on. And then on the x, we have starting from 600 and going to 0. And what it means is that 600 is 600 million years ago, and 0 is the present. And in between, we see how many different types of diverse and number of families we have along the x-axis, the horizontal axis, which is a long time. So how many families of species, how many diversity of things, of life forms did we have here on planet Earth as we understand it from fossils? And there are several things I want to point out in this graph. First of all, you see the 5 black arrows. Those represent mass extinction. It means that scientists went through rocks and dirt and mountains, and during those time, they found a sharp drop in the number of fossils that they find. And it happened 5 times during Earth's history that we know of. There's probably more mass extinctions, but the one that with a lot of fossils in there, where we can see a lot of fossils, and then we see a massive drop in the number of fossils and in their diversity, in the different types of fossils that we find, are these well-known 5 main extinction that started about in the Ordovician era, started about 450 million years ago up till now. Now, what the second thing that is interesting here is that each time after there has been a mass extinction, for instance, after the dinosaur have disappeared and died basically on planet Earth because of an asteroid and because of volcanic activity, we see again a massive rise in number of families, but also in the diversity of the species, which means that in our planet's life, there was a time when there was no ferns, there were no on-land terra plants. And then after hundreds of millions of years, you had these plants. So we see this happening all the time. Groups that were in the ocean completely disappear. And then you find something completely new that's happening on land. So we have this plasticity, and we see that life are able to evolve and are able to be created after mass extinction. And it's especially important because we now realize that in the age of mammals, because of human activity and because climate change is happening, we're going to see again a mass extinction happening of thousands, hundreds of thousands, and millions of species across our planet. But I suspect that as we've seen before in mass extinctions, we will see after this mass extinction, who knows how long it's going to happen. It's going to happen perhaps for 50 million years. Afterwards, you will find a completely new section, new groups of animals, of microorganisms, of plants coming up, living again and occupying this space, okay, of planet Earth. So that's about scale. But let's try and talk for a minute about why does life need this evolution? Perhaps something is created and it's perfect already. Is it? What do we know about it? What do we think about it? I mean, this is the planet. We're here for 3.5, 4.6 billion years. Why does life need evolution? And there are several reasons for this, and I think you already thought about it while I was asking it. Life needs evolution because mass extinctions, and then you need to, of course, to adapt to a new environment. So if first there were very cold temperature, and then something happened, and all of a sudden you have high temperature, you need to adapt to it. Life needs to adapt with it. But there's a very deeper meaning behind this question, and it comes down to why would planet Earth need life forms? Why would something like a life form, a biological life form, will arise from the physical and the chemical composition of our planet? This is not an easy question to answer at all. We don't know the answer to it, actually, but it's a very deep, it requires very deep understanding in order for us to understand why planet Earth needs life and why life has evolved on this planet. I mean, we can talk about it from survival point of view, and then we know, of course, there's new things that happen on the planet, and life wants to survive. Life needs to survive, so they need to adapt to perhaps to higher salinity levels or to lower temperatures. But why, actually? It's a big question, and I want you to keep it in the back of your mind. So what is life? If we're talking about life and evolution, we have to understand what is life now on planet Earth, how we see it today, how it works, in order to understand how the evolution of this happens through millions of years and so on and so forth. So this is a slide that also appeared in our former class, but this, what I want to show you is that it's very, very in brief. So on the right side, you have a microscopic picture that I took in 2013 of a diatom, which is a marine microbial organism. It's about 50 microns in length and about less than 10 microns in width. And the things that are glowing inside it are actually its DNA and where the ribosome, which is an important part of making the DNA into protein and moving into them, working inside the translation from the genetic code into making and creating proteins. That's where it works inside this specific organism. Now, every cell on planet Earth, be it a virus, a microorganism, a mammalian cell, an insect, a plant, has this components like a DNA, an mRNA, a protein, a metabolite, and a membrane, forming it as the cell and making it function. Now, the DNA is the genetic code. It's a deoxynucleotide acid. It's 4 bases, which are connected in pairs, and that's the genetic code. It comes in a double strand with a helical shape, and that's in every cell that we find. Some cells have only one strand of DNA, but then that's the basic. It's a genetic code. It's pretty stable, and all these bases together, they give the code on how to make protein or how to do other things in the cell. Now, the mRNA here in the picture, it's a messenger RNA. RNA is, again, it's a nucleotide acid. It's basically a copycat of the DNA, but instead of a DNA staying in one area in the cell, okay, and being very stable and not moving much, the RNA is the messenger. That's why the small m is there. The RNA comes to the DNA opens up, and the RNA is able to copy the code and take that specific part of the code and take it to a new machinery inside the cell. Now, that machinery, it's not mentioned here, but you're going to see a very short film, a 3-minute video clip in a minute. That machinery is called the ribosome. And the ribosome is where everything happens inside a cell, okay? So the ribosome is a big protein. It has 2 units. And the messenger RNA sits inside, and the ribosome is the one that makes sure to bring amino acid, put them one after another, and then make new enzyme. And that's what the ribosomes do. In mammalian cells, okay, in one cell, we have 10 million ribosome copies, okay, at times. Huge amount of these little machines that the only thing that they do is to create new enzymes, new proteins for the cell to function properly. Now, enzymes, tens of thousands of enzymes in each cell. Protein enzyme, it's like a 3D molecule. It functions. It takes other molecules. It puts them someplace else. It takes 2 molecules. It's put them together. With these proteins, you get a lot of work done in your cell. And of course, another really important component in a life form, which is the cell and how the cell is built, is this envelope thing, a membrane, something that envelopes all these internal machineries and the genetic code and the mRNA and the new proteins and all these things. And this membrane is what we call permeable, which means some things can go in, some things can go out, but some things are blocked inside. And it's really important for life if you want to, protect it from the harsh outside, whether it's UV, whether it's radiation, whether it's high temperature. This membrane can be very thick. It can be very successful in blocking a lot of those harsh conditions outside. So that's like a basic component of a cell. And what you see here is ribosome in action. The ribosome structure was elucidated and worked on by Professor Ada Yonat, who was a Nobel laureate for 2009. She works at the Weizmann Institute in Rehovot in Israel. And what you see right here is the messenger RNA. It's a beautiful animation, and I wanted to show it to you because the ribosome is so important to understand how the mechanism from the genetic code goes into enzymes and how it's relevant for the evolution of the organism and the evolution of population on planet Earth. So basically, this is the ribosome. Okay, the ribosome itself is encoded by a genetic code. It's very conserved. And what we see here is that we see another type of RNA, which is called transfer RNA. And this transfer RNA, it looks a bit like an L shape upside down or like a T shape. This little thing brings at the end, at the very bottom of it, it brings an amino acid. And it knows how to put the right amino acid one next to another. And you can see it at the bottom of the screen here, at the bottom of this video clip. And what you see is a new line. Now, this is a new line of amino acid. And what is not shown in this video is that this new line, it then folds upon itself and produces like this new protein. It's an amazing process. But this is the ribosome. And this is where basically the integration between what is in the genetic code and what's happening later in the cell with protein, this is where the main mechanism happens with this ribosome, which has 2 subunits. Now, from that little complicated, complex mechanism in every cell of planet Earth, we have this huge amount of life forms on our planet. Just like billions of life forms, millions of species, and I just wanted to give some sort of a touch to it. So on the left side, you see a picture, an image of planet Earth, and it shows in red where you have concentration, the highest concentration of terrestrial, so it means only on land, vertebrate species. So vertebrate species is a class, is a group of organisms on planet Earth which have bones, okay? So I have bones, giraffe has bones, insects don't have bones. Not only mammals have bones, but other species have bones as well. So those are just the vertebrate species. And the vertebrate species are actually not the biggest, aren't the biggest taxonomical group in the whole species of the world. We have much more insects, we have much more microorganisms. But this to give you some sort of a feel on how we look at these things today, and that we're able to say, okay, across the equator, we have the highest concentration of diversity, the highest number of different types of species of vertebrate terrestrial species. And of course, we have a few in the oceans, they're not shown here. And on the right side, we have all these little picture which shows so much that has been collected from some forest, and it's just the mushrooms and the lichen, and all that diversity started from that mechanism that I showed you before, that you have a genetic code, and from the genetic code, you get eventually a protein and other stuff and how the cell should function. So what is the connection? How does evolution works? So what is evolution? So evolution is the process by which organisms change over time, right? Mutations produce genetic variation, so the code changes in population. So it's not just about one organism, it's about its whole group, like thousands and thousands of species of human, thousands of species of insect, thousands upon millions upon billions of microorganisms. We're talking about populations of those species, big numbers. And the environment interacts with this variation to select those individuals which are best adapted to their surrounding. Now, this sentence came from Darwin, and we're going to talk about it in a minute, and it's called natural selection. So the natural selection says not everybody gets to evolve. A lot of people go through mutations, a lot of species go through mutations, but the environment interacts with this variation, and only those individuals who are best adapted because of this mutation will go forward, okay? They will leave behind more offsprings than less well-adapted individuals. So if you give it enough time, one species may evolve into many others. And the hardest part for me is always to understand what is given enough time, because our brains are not really capable of understanding what it means to have millions, tens of millions, hundreds of millions years for this process to occur. Okay? So if we go forward, I want you to think about evolution as something that happens in time and in space. And we know it from planet Earth. So you have the entire planet as your game board to do all these changes. And some changes that happen in one section of Earth are good for a different section of planet Earth. But these things are all the time bubbling, and they're happening, and they go through all time phases since the beginning of time. It's a continuous process. It never doesn't happen all of a sudden. It doesn't appear and then disappear. It actually happens continuously across the entire space and time that we have here on planet Earth. And when we talk about how evolution came to be and how we understand it, there are three main figures, but not only, there's actually more. But these are the three main figures which are absolutely essential in order to understand what is evolution and how we got to where we are today. So from left to right, we have Jean-Baptiste Pierre-Antoine de Monnet, Chevalier de Lamarck, or in short, Jean-Baptiste Lamarck. In the middle, we have Gregor Johann Mendel. And on the right side, we have Charles Robert, which probably you have heard about it. And the year beneath the picture is the year when they published their most, their most significant scientific work or book, in which they describe what they think about evolution or their big finding. So Darwin was in 1859 and Mendel was in 1866, but they didn't know about each other. Charles Darwin was not aware of what Gregor Mendel was doing. And of course, Jean-Baptiste Lamarck had a completely different view of what is evolution based on. I did not. It's working on my side. I'm looking at myself.
Okay, I'm not able to see it somehow. So I'm just going to take you out of the screen so that only the presentation is visible. And maybe
Sure.
when I can see you again, I'll bring you back in.
Sure.
Okay, I think we have established the connection now. You can start again.
From Charles Darwin?
Yeah.
Got it. No, not a problem. Technical issues is my favorite.
Yeah, it always happens.
So not a problem. So Charles Robert Darwin was an English scientist, and in 1859 he published the Origin of Species book, in which he talked about evolution and other issues, and I will go into that in a minute. But Gregor Johann Mendel published his work in 1866, and yet these two smart gentlemen didn't know one another, didn't recognize each other's work, which is a shame because they were working in very close areas to one another, and they could have used a lot one another's help in regarding to their own ideas and scientific thought. Lamarck was much earlier, and he was talking about evolution in different terms than Darwin, and I will go into that in a second. So what did Gregor Johann Mendel did, and why he's such a father figure in genetics and in biology? He was a prior for an abbot. He was a friar and an abbot in a church in what is today the Czech Republic, and back then was the Austrian Empire. And he did a lot of work with peas. He basically took green peas and then yellow peas and then big peas and small peas and lengthy beans and started doing a lot of crisscrosses. So he did what they called artificial selection. And what he realized is that something inside the peas goes inside each generation and sometimes skips a generation. So sometimes the green color would emerge in the next generation of the crossing of the breeds, but sometimes it would skip a generations. And he was able to understand that there is hereditary things that are coming from inside the peas and have nothing to do with the environment, have nothing to do with the soil or the time of year which he breeds them, but there isn't a hereditary something going on that also affects how the peas will be later on look and taste, and what are their size and what are their colors and so on and so forth. So some of the basic ideas in genetics, such as recessive genes and dominant genes, come from Gregor Johann Mendel's work. And that's an amazing thing that he came up with in the 19th century. Now, Charles, why doesn't it? Yeah, so Charles Robert Darwin, or Charles Darwin as we know it, was an amazing English scientist who came up with his species of origin in 1859. And he understood, after being in the Galapagos with His Majesty's ship, the Beagle, I think it was 1836, and he walked around and he documented all the life forms on that island. And what he noticed was that there are finches, which are small birds, and he noticed that there was a group of them with different shapes and size of beaks. And then he realized that what happened on the island is that these finches, along many, many years, each different finch has adapted to eat something specific else from other finches. So when he looked at it, he said, this is natural selection, which means in order for all the finches to survive on the Galapagos Island, they had to adapt to their environment. And those that had the right mutation, and for instance, had longer beaks in order to find worms, or had other beaks in order to crack seeds, these were the successful one. And eventually the population of the finches in the Galapagos Island was diverse enough to issue every ecological niche so they can have what they need in order to eat and to reproduce afterwards. And from then on, he jumped into the main idea of the natural selection, in which natural selection is a process in which mutations happen all the time and changes are caused over time. But only the individuals which have a successful mutation, which helps them in their environment, in their physical environment, those will have offsprings. And those offsprings will have the same criteria, the same trait that will go forward to their own offsprings, and so on and so forth. So that is a process that is called natural selection. It happens naturally. Either you are okay with your mutation and you survive and you bring offsprings and you reproduce, or you die in the process, or you're not as successful for food sources as the other guy who has a bigger beak. So that is the basic for the evolution in the world as Darwin thought about it when he published his book in 1859. Lamarck noticed also changes in the environment, and not in the environment, changes in biological beings, in life forms, but he didn't think the same way as Charles. Lamarck's way, and now it's known as Lamarckism, has a different way of looking at it. So Lamarck would go and say something like, okay, there are two forces happening. First of all, there is this issue of whether the organism is using correctly, the tools that his body has. So if he's using and needs a lot of muscle in the left hand, then the left hand will be overdeveloped, and that's how mutation and that's how evolution will go. It will go per an individual, it will go per an organism in that individual. And then secondly, he said everything goes from simple to complex. So the ideal is a very complex system or complex organs that sustain this life form in life and makes it very, very successful. Lamarckism didn't talk about hereditary, didn't say anything about offsprings. He really believed that the organs during the lifetime of an organism can develop and move and flourish and be exactly to what they need during their lifetime during their lifetime on planet Earth. So it's a bit of a difference than Charles Robert Darwin. It's not a natural selection. It's a different type of process. It happens per organism. It goes from simple to complex, and it has this adaptive process that goes while the individual is alive. And Charles didn't think it. Charles just thought mutations happen. And if they're good, then that individual survives. It starts from somewhere else. It doesn't start within the lifetime of that individual. So these three individuals are really, really huge, because if Charles would have known about the genetic heredity that Gregor Johann Mendel thought of, and he didn't say genes. Gregor Mendel did not find genes. He just find this issue of hereditary, that something comes from parents to children and from children to their children. He actually found the different traits. If Charles Darwin would have known about the hereditary that Gregor Mendel has been, scientifically analyzing, then he would have found exactly the thing that brings up and, makes natural selection such a big idea. Lamarckism is not completely wrong. There's something we now know that is called epigenetic, and I will talk about it later. But when we're looking at what is evolution and what is the basis for this biodiversity, so on the right side, Darwin said, look, someplace long time ago, there was an ancestor finch. There was a common ancestor to all of the finches that I see now on Galapagos. And from that ancestor finch, there was a natural selection process, and I got different type of finch. One was insect eating, the other one is a seed eater, and so on and so forth. And you can see this natural selection concept, going very well with other things that we know of. So some things would look much more attractive to their mates than others, and then they would grow and they would reproduce much better. And the same thing that goes for finches for sure goes for other animals and insects that eat, and that's a lot of life forms on planet Earth. So what Darwin thought about as the common ancestor was a really, really big discovery, actually. And you can hear the audiobook of The Origin of Species, and I recommend that you find it on the internet because it's a beautiful book to listen to. It's written very nicely. So how do we go from this natural selection with beaks and eyes and colors, and we go back to the genetic code? And that's where I want to go next. On the left side, what you see is the double helix of the DNA rotating around, and we want to work with it with the messenger RNA and so on and so forth. It needs actually to break open. So what do we know about, and what is evolution, and how do we look at it today in through scientific eyes? So don't try to read what's in the right side in the right pane, but that's how publication goes on today. But on the left side, there's a simplified version of what's happening on the right side. And that's called the tree of life. And that's how we see evolutionary process today, and how we talk about it through phylogeny. So on the left side, we have basically what we see. We see, like 3 colors. We see above them the names bacteria, archaea, and eukaryotes. And these are known as the 3 super kingdoms of life. We have a lot of species going into bacteria. The difference between bacteria and archaea has to do with their DNA, has to do with their membrane composition, but both are microorganisms. There's no such thing as hey, archaea that you see on the street. That doesn't happen. Archaea and bacteria are massive, millions of life forms, billions of life forms and millions of species, but they're microscopic basically. But eukaryotes are different. Eukaryotes have the 3 kingdoms within them, which is plants, fungi, and animals. And this is something we're more familiar with. And the main difference, if you ask microbiologists and molecular biologists, what's the difference between eukaryotes and the rest, is that in eukaryotes, inside the cell, you have additional smaller organelles in which you have the DNA and the mitochondria. In archaea and bacteria, you don't have additional organelles or additional things inside the bacteria and archaea, and the DNA usually goes inside the protoplasm, free, inside the cell itself without additional structures. That's the main difference between bacteria and archaea and eukaryotes. But what you see beneath this nice rainbow of species and a little bit of names that you see there is that you see these squares and you see these lines, and they're clustering together, and those lines are getting together to one single line. And what happens is that we were able, in the last, 40 years, to take the genetic code, okay, and then understand what is the relationship between the genetic codes of all these different species. And according to the differences in the genetic code, we can assign them not only to different clusters, but also we can assign the length of the line, okay? The length of the line is representative of how far away and how much changed the DNA, how much mutations are in the DNA from one species to another. So it's like a length system, basically, and it's a phylogeny tree. And it helps us sometimes to understand in a 2D representation what is the relationship between the genetic code of all the things that we see on the tree, and also how far away they are from one another. And we are able today actually to put something which is called a molecular clock. I'm not going to go into it, but we know if some species has appeared, let's say, 450 million years ago, we can assign a certain time lag to its mutation, and then we can understand that another species, which this first species was its common ancestor, it appeared 250 million years afterwards. So this is how we look at evolution at the moment. This is called, the tree, phylogenetic tree. There's a lot of algorithms that go into it. I'm not going to go into those algorithms at the moment. Again, I think Wikipedia has done a magnificent job in going in depth of this and in a very simple term. simple terms, feel free to go to Wikipedia and check out their evolutionary and biodiversity. Now, as we go through the genetic code and we do the modification, other things happen in the body. The beak becomes wider, the color of the eyes changes. The genetic code is responsible for some of those changes. So it's super interesting to see how from genetic code we go into this issue of evolution. And it's interesting to see that already in 1837, Darwin, in his own personal notebook, had this tree, scribbled. And he said, I think, above it. Because he understood it's a great representation to understand, I have all these finches, but I think they have a common ancestor. So all the branches go back into a central branch. And that central branch is actually part of another group of different branches, and they go to another central branch. So all this is an excellent representation of things that can disperse if they started from one origin. And one of the things as molecular biologists is that all of a sudden we find sometimes that we have things which do not come from one origin, and we find mechanism in which you can hop a genome from one species to another without going through a common ancestor. So there's a lot of details and a lot of funky variation that goes into this. There's not only just one dogma, but it's amazing to me that from the 19th century till now, we're still working with phylogenetic tree, and we're still working with the minds of Darwin and his colleagues. It's quite an amazing thing to think that we're working now for 200 years on this, way of thinking. It's amazing. So if we're just going quickly into a brief summary of what we talked about so far in this presentation, and I'm sorry for the technical issues if they were, is that we discussed when life began, and I told you that we now believe that it's between 3.5 to 3.9 billion years ago. We talked that it might have started, in hydrothermal vents and so on. We talked about time scale. Basically, 600 million years ago, that's when we see fossils. Before 600 million years ago, we did not see any fossils. And because of that, we think that before 600 million years, they were mostly perhaps microorganisms, or a lot of organisms which didn't have any carbonates, didn't have any skeleton, didn't have any bones, didn't have perhaps other multicellular, didn't have tissues, complex tissues to leave marks on rocks, and we don't find them. So actually, multicellular life is something pretty big, but it's only for the last 500 million years old in our planet. We also had mass extinctions there. We asked the big question, why life undergoes evolution? And from a survival point of view, we know life needs to adapt because there are mass extinctions, because the environment change, and life is always propagating to survive, and it needs the change. But one of the things that Darwin found, and it changed our life, is that it's not that life needs to go evolution, it's that mutation happens all the time. And it's just the selection afterwards of the environment that keeps up with all these diversity and things that are adapted to their specific ecological niches. We talked about what is evolution. We talked that it's a change over generations, over time, for entire population. We talked about the selection with Darwin. We talked a little bit about Lamarck, about Darwin, about Mendel and his genetic work. And we also talked, and I tried to explain as best as I could, about the natural selection by Charles Darwin, having a common ancestor and all these mutations happening until everybody fits to their environment if given enough time. And I also showed you what looks like evolutionary trees. So this is like a 2/3 summary, and we're almost at the end of my quick presentation. So how does molecular biodiversity evolution looks like today? So I want to put this aside at the moment and talk to you and let you know that for many years, microbiologists and biologists in general, they would look at how things look like, and they had a connection between the shape of the leaf in different plants, the shape of the stem, the shape of the flower, sizes, colors, and so on and so forth. We didn't have any of the tools to look at the genome at all. We had tools like Mendel used. Peas, we crossbreed them. Corn, we crossbreed them. So we had to look at the traits before we looked at the genetic code, and a lot of the taxonomical issues and how things are related to one another came from our eyes and from scientists who 3 centuries just looked at things and made a very detailed analysis on how things looks and what they look like, but they didn't have any access to the genetic code. And what happens in recent decades is that with the genetic code, we had a lot more understanding and some big changes in how things are related to one another. And at the moment, I just want to show you how I work, how molecular evolutionary studies work like. And I work in extreme environments, so I'm very much aware of these, mutations and how they express themselves in order to make a organism very adaptive to its own environment and be successful and reproduce. So basically, when we work today in molecular biology, we take a sample from something that interests us. And it could be an organism like here on the table with all these fungi and lichens, and it's awesome. Or it could be, I want to do a metagenome study and say, I'm not interested in just one organism. No, I want to see what's in the soil of a forest, what's in the sand in a hyper-arid environment. And then I just take the soil as is, or I take the water, or I take the, lake water. I take everything that I need, and through a lengthy process, which includes a lot of chemical steps and physical steps, I basically extract the DNA from that sample. The entire DNA in a sample goes into a test tube, and eventually, again, with a lengthy process with chemicals and physical processes and so on and so forth, and at least 2 labs and maybe a week or 2, and eventually what I get back is very simple. I get a text file. It could be 5 megabits, it could be 70 kilobytes, it could be 1 gigabyte, but it includes thousands and thousands of sequences that looks exactly like the one on your screens. So you have the 4 bases of the DNA, G, T, G, C, C, A, in a text file, and you have thousands of those, each one starting with a unique identifier, like the one here, which is MN01106. What do I do with this sequence, with this text file, basically? I can do many things with it. At the moment, we're focusing on two things only, okay? What I do with it is I can do 2 things. So the upper arrow, basically what I do is that I take all these sequences and I make an alignment. I put them one underneath another, and we're talking thousands and thousands and thousands of sequences. And we have now various programs which know how to run and do an alignment. And what I'm looking for is 2 things. I'm looking for 3 things, actually. I'm looking, one, where all the position in which the code never changes. So you see, for instance, there's a black G, right? And there's there are 2 nucleotide bases, and then there's another, like a column of black G. That means that in that specific alignment, the G's never change. So I'm looking for conserved genetic code areas. Could be areas, it could be just 1 letter, because 1 letter is important in our genetic code, and I'll explain in a minute. I'm looking for areas of diverse. Things change. What is the meaning of the change? Does it change? Is it a coding area? Does it change the protein? Maybe it's negligible, maybe it's not a mutation, maybe it just changes and it has no meaning whatsoever afterwards. We don't necessarily know. And the third thing that I'm looking for motifs, and I'm not going to look into that, but in our genetic code, we have areas that are repeaters, and they have significance for RNA, for other proteins. They have a very significant, it's like a slogan, it's like like a password for a protein to recognize this sequence on the genetic code in that area and come and work with it if needed, okay? So 3 things, the conserved, the not conserved, and motifs. So that's the first thing that I can do with these thousands and thousands of sequences that I get. What is more interesting to do, and that's what I'm going to go now into detail, is what the lower arrow is pointing. And the lower arrow is pointing at a table which includes triplicates, triple of the AGCT code. And what's happened is that since 1961, scientists have realized that every 3 letter in the genetic code, okay, represent 1 amino acid, okay? 1 amino acid, which means if I have, for instance, a TTT triplet in the genetic code, at the end, after the ribosome that I talked about before, at the end, I will have amino acid which is called phenylalanine. If I have a triple of, let's say, U, then I'm going to get a different amino acid. And this is the big thing, because from the genetic code into amino acid, into the protein, it means that we know which regions in our genetic code are coding for actual proteins. And proteins is what keeps us alive. And for instance, when you take antibiotics, they usually target different proteins. For instance, the ribosome, and they stop proteins being made, and they, hit the microbe where it's hurting him. The microbe doesn't get the protein that it needs. No proteins are being created because the ribosome is blocked, and the microorganism die. That's how most antibiotics work. So we have this triplicate issue now. So we're interested, again, programs, it's automatic programs. They take the sequences that I have acquired, and they start going through all the possibilities of what type of amino acid. And then they rise it up, and they tell me what type of protein it is. And then I know which function in the cell I'm looking at. Is it a protein that's involved in photosynthesis? Then perhaps it's about the evolution of this microorganism to do photosynthesis in a salty environment. Perhaps it's something else. So for instance, if we take into consideration here, it's a work I did a long time ago, and I have these sequences. What you see now is an alignment of amino acid. Amino acid can be represented with a one-letter word. Here, one letter, sorry, not word, one letter. So for instance, you see here an alignment. We start with CDPKA. So CDPKAD, for instance, it's cysteine, it's glutamate, it's proline, it's lysine, it's alanine, and it's either glutamate or the other one, or glutamine, for instance. So I know this is a protein. And today, because we have so much computer power and we have so much knowledge about the structure, the physical 3D structure of proteins, I can take this alignment, I can say what areas are super conserved, just like I did with the genetic code, and I can say which areas are not conserved, just like I did with the genetic code. The areas which are not conserved are the areas where we will see mostly mutation, and we'll see the adaptation to their environment. And then I can interlay this, I can put this, I can do an overlay of this information, this evolutionary information, and I can put it on a 3D structure. And what you see down here basically is a 3D structure of part of a nitrogenase protein, which is relevant for nitrogen fixation. The maroon purple area are mostly conserved, which means doesn't matter how many sequences I will try, this area will never change. It will never mutate. Evolution will never change it because it's perfect for its role, and it's vital. If you change one amino acid, it won't be able to catch the molecule that it needs. It won't function, it will die. So for instance, if I am a pharmaceutical and I'm interested to nail down this area, then perhaps I should look at this area, and perhaps I should design a medicine or an antibiotic or a molecule that can go straight into that area and block. Okay? One of the things we do with medicinal thing is that we're looking for these different areas in a protein that now we know how to analyze, and we're looking what will be the best place to put a medicine or a molecule that will block this protein from functioning. And it's a big issue. It's not easy to find it. But just to give you an idea how we go from sequences to function and to this structural area and to evolution. Okay? What we see when we talk about adaptation for evolution today is that we start from the DNA sequence and it has mutations, right? That's what Darwin said. There's a natural selection going on. Those mutations change the sequence of the protein because we now know that triplicates in the DNA, they go for amino acid. You put amino acids together, you get a protein. From the protein of its sequence, you get the structure because those amino acids, they're molecules. They twist upon themselves and they connect with one another and they send electrons to one another. So in change, you have adaptations for evolution that starts from the DNA and end with a different protein. And once you have a different protein, the sky's the limit. So the beak can change because the change is not just in one protein, it's in thousands of sequences of DNA that goes and codes for thousands of protein that need to work together. So the color of your eye would be green, or the shape of the beak for this finch would be wide to eat seeds and would be tough. So when I'm talking about space and time, I'm talking from the very small scale how mutation occur in DNA and then how it affects the physical world, which is huge, the world, planet Earth that we live in. So it's an amazing process. It's hard to grasp if it's the first time that you hear about it. It's hard to grasp this. But this is how we look at it when we look at molecular evolution nowadays. From the genetic code but like multiple, like many times the genetic code situation, repetition, mutation, and things like that. Some of the mutation are really, really bad for the individual, but some of the mutations are actually really, really good. So what happens when you have, hundreds of mutation happening? Do the effects sort of like goes average? Do the effects, completely cancel one another? It's a big, big issue. And then until eventually you see it in the individual, in the organism. That's how we perceive it today. Now, if the finch is happy and it eats well, then it will reproduce better. And all these changes in the DNA, they would probably change and will move on to the offspring. That's what basically Darwin has suggested. However, there's a reason I put these arrows in two directions, if you'll notice, because we found now with a scientific area which is called epigenetics, we find that it actually goes both ways. Sometimes the environment brings effect to the physical body, which eventually enters into your genetic code and then reproduce and then goes on to your offsprings. So it's a two-way street, and the connection between them is a really big study area for a lot of scientists in the world to understand how much of the environment is affecting the individual versus how much the DNA, and what is the interplay and what is the mechanism. I mean, how does it work? How much of the environmental signal do you need in order to change your DNA? Not a straightforward answer either. And different organisms have different mechanisms as well. So my mechanism is not the same as an E. coli. A fly would work different than a bird, and so on and so forth. So it's a lot of variations, a lot of play into the space and time of how you do evolution on planet Earth. So what about evolution in other worlds? Well, we don't know yet. We haven't found life forms elsewhere. However, it does seem that the physical concept of time works the same way everywhere in the universe. I mean, we can talk about the possibilities in black holes and what happens in their event horizons. But generally speaking, I think time as a physical constant, as a physical, not constant, as a physical dimension seems to work the same way everywhere in our universe. So perhaps it will work, evolution will work more or less the same from a time perspective on other exoplanets. And again, I'm going back to the really deeper question on why would a planet need life? What's the purpose of life forms on this planet? And if there's a purpose, which is a very dangerous word to use because it means that we're using our human minds and trying to find our human needs and human goals on something as big as a planet, which basically has nothing to do with our human brain. I mean, why do you need life forms in a planet that has, it has a revolving core, has geochemical cycles, has sulfur and nitrogen, and it has oxygen in the atmosphere, and have an ocean to swirl around? What is life form there? Why would they arise at all? And if we know why, then we can answer a really big question. Is it a universal process? So if time as a physical dimension is the same everywhere in the universe, is the emergence of life the same thing? Is it a universal, known factor, process, constant? Who knows? We don't know at the moment. It's a great big question. And in my opinion, there's a potential is huge for amazing variations of life forms. We basically just scratching now the surface of life forms and evolution on planet Earth. We just now basically realize it as humans, as human species. And I think, therefore, there's a lot that we don't know, and therefore the amount of variation that can be out there is quite mind-boggling. And I would be very happy, for instance, if Perseverance, that would land in, I think, February or April 2021, would find more probably more robust, more better evidence for past life on Mars, because then we can really come to the question that perhaps it's a universal process. Perhaps other planets, life starts from the physical environment. Until we get those evidence, until we see more evidence, more facts, more analysis coming from other planets, we just don't know if our planet is unique. And for those of you who were able to stay till the end, first of all, the summary. So the third part of my lecture today was about how we do molecular evolutionary study, how we look at the genetic code, what are we doing with the analysis between the DNA, mRNA, protein analysis. There's a lot of more things going into it, which I haven't spoken about. That's fine. Just, check your sources. I'll be happy to answer what I can. Adaptation can happen on so many levels. So that adaptation can happen on the genetic level, on the protein level, on the membrane level. It can happen as a population. It can happen in your environment and affect you back. There are different layers here of adaptation and adaptation techniques for the individual. And we don't know about other worlds. And I would like to show you this riddle just for the end. If you managed so far, well done. And I'm just showing you this image. Okay, so you need to figure it out. And thank you very much.
Okay, guys, you can just figure this riddle out and then you can send us the answers. We will check with Reut and we will see how those answers are correct. Reut, is that okay?
Yeah, that's wonderful.
Okay, perfect. So do send us the answers on support@astronera.org if you'd like it. Or you can also comment on the video, so that other people can also see and learn what the riddle is about. Anyway, Reut, can you stop sharing your screen so that we can have a chat and have the questions?
Yes.
Okay, perfect. So let's begin with the questions, but before that, I'd really like to tell you that this is such a niche topic, and of course, we are just still scratching the surface, and there is a lot to know. And just yesterday, somebody was asking me about the Darwin's evolution when I was doing this Ask AstronEra talk, and today you gave like perfect elaborate explanation in terms of astronomy as well, so that was really awesome. And I'm really hooked to this session. Like from next time, I'm gonna sit with pen and paper and write my own notes as well. Yeah, and with the perseverance as well that you mentioned, I hope that we find some evidences on Mars, and it'll be really interesting. Maybe next year you can talk about the astrobiology from the perspective of Mars, because right now we only have the Earth to we cannot have anything else to compare the life with, so we have only Earth as our example. So yeah, that is really tough, but the way you explained everything, it was really great. Okay, so diving into the questions, Adarsh, Adarsh Kumar has asked, how are stromatolites extracted? So stromatolites, as you mentioned, are the oldest still record. So how are they extracted?
So the stromatolites that live today at Shark Bay, you need to get permissions, and then you can go and you can work on them together with the Departments of Environmental Conservation in Australia. You can find stromatolites in other places in the world. I think you can find one in Canada and in China, and you can work on them and extract the DNA from them. And the way to work with them, so in order to get DNA out of rock, you basically need to work with a DNA extraction in which, first of all, you pound the rock and make it like a powder. Make sure that it's very, very, how do you say it in English? But it's powder-like. It's more like a dust than continuing as a rock. And then part of our protocol for the DNA extraction is to use EDTA, which are chelating factors, and also using other things which will leach out the carbonates or other minerals that are within the rock and would leave the DNA intact. And later on, what we do is that we use centrifuge, and with centrifuge, we downsink the heavy particles, which are the minerals and salts and carbonates. And then we just are left with other lighter things in the liquid in the centrifuge tube, and those liquid go through additional processes of centrifugation and other issues in order to make sure that what we have in the tube and we send to sequencing is just the DNA, no protein, no membrane, no enzymes, no anything else that might interfere with the sequences. I hope that's a good answer, if that's what you were looking for.
Yeah, that works for now, I guess. So we have question from Rupa Dhavre. How life shaped the earth and how earth shaped the life. That's a very elaborative question that you will have to answer. Do you think you can put it in like some small?
Yes. So I can give one example. For instance, when there was no life, a lot of the iron would be oxidized by the atmosphere. And so you would have a lot of red areas. So, for instance, what we see today on Mars is basically because there's no life on the surface of Mars for many, many, many years. On planet Earth, you have things which work with the upper soil, plants, fungi, microorganisms, and they use the iron. So instead of oxidizing and getting a lot of, for instance, red color in soils and mountains, we do have different colors and shapes on planet Earth. Life, that's a good example on how life has influenced planet Earth, for instance.
Okay, perfect. So again, a question by Adarsh Kumar. What are the major reasons of mutation in vertebrates?
I don't know what is the main reason for mutations in vertebrates. I know there are different things which cause mutations. They could be free radicals in the cells. I mean, molecules which, for instance, have a free oxygen, and that's a very radical, it's a very activating situation in which the oxygen takes electrons and then can harm the DNA sequence. I know UV and other radiation types, gamma radiation, radioactive radiation, they cause breaks, and because they cause breaks in the genetic code, the genetic code have a way to fix itself, but sometimes it fixes it in the wrong way and puts inside, for instance, a different base than it was before. And so this type of mutation goes into the genetic code and multiply, and if you replicate the genetic code, this type of error actually multiplies as well. So that's another type of accident that's happened in our genetic code. A lot of problems in our genetic code eventually cause cancer, as far as I know.
Oh, okay. All right. So we have question by Federico. He's asking, how would organized beings such as us came to being in a universe that tends to be so disorganized?
That's a great question because it points out to the disorganization or the chaos in the basis of our universe. And what we do see, Frederick, is that the universe really likes to lower its entropy and get things in order, because once things are more in order, their energy level lowers, and if I'm not mistaken. And so what happens is that one way to look at life forms is to look at life forms as something which organizes the universe even further. So you know how first there was a big bang, and then eventually a few gas clouds, and you had all of a sudden you had the sun, and the sun started getting all the dust around it and all the things until it started working as a nuclear powerhouse, and then you have the different planets form. So there's this movement in the universe into organizing things. So I think life forms, all life forms, not just humans, are some sort of a process of trying to put order into disorder.
Okay. So again, you answered the question by Adarsh Kumar before, and you said that we could powder the stromatolites, but wouldn't it affect the DNA strands that we ultimately want?
What would affect them? What would affect? Again?
The powder.
The powder from the rock would affect what again? Sorry.
Okay, so he's asking that if we powder the stromatolites, doesn't it affect the DNA strands that ultimately we want?
No, DNA is not affected by the powdering process.
Okay. So we will take this one last question for now. Samarin Ansari is asking, can we separate stromatolite by PCR technique?
Okay, so PCR is something which is very relevant to the presentation I just did. And I didn't go into it because we didn't have a lot of time and I wanted to focus on evolution and not on all the processes. So PCR stands for polymerase chain reaction. It's a step after DNA extraction. After I do DNA extraction, whether it's from stromatolite or from soil or from wood or from whatever, PCR is a process in which I amplify the sequences of DNA like a billionfold. A billionfold. It's a thermo reaction. It goes through different temperature, and I'm able from one strand of DNA to get like 30 billion strands of DNA, which I then put into a sequencing machine. So I think what he asks here, Kumar, is Samreen Ansari, is that whether we use PCR in this process of after doing DNA extraction for stromatolites, the answer is yes. But it's not like stromatolite have their own, group as sequences that I can do a specific PCR that would be a stromatolite PCR. Mm-hmm. So sometimes we do PCR when we want to target a certain gene in a certain group. So I know exactly how, an E. coli gene is, and then I amplify just that gene from the entire DNA pool sample that I have. Mm-hmm.